Hello everyone, welcome to my Statistic YouTube channel. Today I am going to demonstrate the GATK or GATK tool and its best practices for genomic variant analysis. So in first step, I'll demonstrate how to install the GATK tool, and after that, for the genomic variant analysis, first we'll do the add and replace read group of the BAM file, and after that we'll then use the mark duplication for our BAM file for the genomic variant analysis. So we have the 6 BAM file here. This BAM file are the sequence for 3 without ethanol treatment sample and 3 10 ethanol treated sample. I made this BAM file from the various steps. You can see my video for this the previous step. So here I will discuss and dictate and demonstrate how to use this BAM file for the GATK tool and for variant analysis. So for that, first we have to go Google and search GATK tool. So go to this site. So go to the download latest version of GATK. So here I am going to download this zip file. So it is 671 MB of tool. Okay, so the download is complete after one failure, and let's go to it. So let's extract. Okay, extraction is done. Go to this file and open terminal. So just ls l. So here you can see it is a jar type of package and pre-compiled, so you don't need to install it, but make sure that your system has Java installed in latest version. You can check out my Java installation video for your help. So let's check it as Java -jar, then the package local, and let's check it, enter. So here you can see different type of GATK tools is here. So for variation analysis I am going to use AddOrReplaceReadGroup for the first step. So in the variation analysis we have 6 BAM sample, and this BAM sample are in two group, without ethanol and with ethanol sample. So we have to run the add and replace group tool for each of this BAM file. After that we have to run the mark duplicate analysis for all of these BAM file. So first we have to do the add header. So add and replace read group actually add the header for each BAM file in our analysis. So to do that, let's copy all our file to this directory. Let's check it. So it is java -jar, then the package local add or replace read groups. Let's enter. So here we can see the argument and usage of this tool. Let's copy this one. Just have a note. So here we can see the usage is first we have to one input, there would be an output and read group ID and some other arguments. So these are the required arguments: input, output, and this is read group library required, read group platform. So let's do it. So let's make a script here to add or replace the read group. So first I have to copy this one, java jar, and put it here. Then I have to use the input. So let's input. So for input file I have this one, this 83. Let's copy the total name and let's paste it here. So the next is output. Let's copy it and paste, and so copy this one and paste, and I'm going to add this file as a add or replace read group name. Okay, then you have to write the LB. Copy this and paste it. So LB is read group library. So it is like ethanol stress, ETOH stress. Then I have to put the PL. Let's copy it and paste, and it is the read group platform ILLUMINA. Just copy this one and paste it here. So in next we have to use the PU. Copy and paste. So in PU you can just use this one, unit 1, and paste it here, and after that you have to add the SM. Let's copy it and paste it here. So SM is the read group sample name. So here the sample name would be without ethanol. So here I have to just add this, copy, replacing this one, enter. So it is a total script to add or replace the read group, copy this and just clear it, and I'm going to paste. So let's enter. Okay, the run is done. Let's check it. So here you can see the output file is generated. So you have the script to run it for all these file. You can use bash script for this. Let's copy it and make a script. Let's empty it is add or replace read group dot sh. Just open it and place it. So here you have to just write this one and just name it local, and the local will be this jar file. Let's cut it and paste it here, and you just use this one, copy and $sign and local. So it is the script for the 84 sample. Just replace in 84. This is 84 and all are same because the 84 is also the ethanol without ethanol sample, so it is fine. And do it for the 85 sample. So just change it to 85 and 85 and control save. So after writing the script, just save it and close it. Now just check the list. Just clear it and list. So here you can see this is your script. You have to run this script to analyze these two files. Okay, so to run it, write sh, then this script and enter. So in this process, it will first run this script, and after that it will run this script. Okay, it is done. Let's check it. So you can see for the next 3 BAM file, which are with ethanol treatment file, let's do it. Let's copy it 3 times. First I am going to add hash to block this, so it will not run this command and this command. So here I have to change this BAM number, like here is 89, 90 and 91. Let's change it. So it is with ethanol sample. So total script is ready. Just save it and close it. Let's clear it. and run the same script and enter. Okay, so the run is done. Let's check it. So here is the file. So in the next step, you have to do mark duplication. This video is going quite long, so follow my next video for mark duplication process. If you found this tutorial helpful, please give it a like and leave a comment. For more bioinformatics tutorial, don't forget to subscribe to my channel. Thank you.